Hello students, welcome to the fourth and the last lecture of the four lectures on glycogen, glycosaminoglycans and glycoconjugates. I think this particular lecture will focus mostly on the pathobiochemistry of some of the disorders which are associated with glycoconjugate metabolism. And unfortunately, you have to remember some of this information because they are sometimes or most of the times they are inquired about in the assessments. So they are very, very important. We start with mucopolysaccharidosis. These are rare inborn errors where there is a problem in the degradation of glycosaminoglycans that result in a series of diseases called MPS. And the key aspect of these diseases is that they are associated with mental retardation and structural defects, and in most of the cases it is both. That means there is both mental retardation and structural defects that are observed. They have been classified into MPS 1, 2, 3, 4, 6 and 7. Unfortunately, there is no MPS type 5, and we need to know some of the disorders in a bit of detail, especially with regards to what is happening in these disorders. So we have MPS type 1, which is commonly known as Hurler's syndrome. It results from a deficiency of the enzyme alpha iduronidase, and this particular enzyme you need to remember. And because of the deficiency of this enzyme, there is an accumulation of heparan sulfate and dermatan sulfate. The phenotypic features are associated with severe growth and mental retardation. And especially in this phenotype, there are characteristic changes in the facial features of the patient. Next, we come to MPS type 2, commonly known as Hunter's syndrome. It is similar to Hurler's syndrome, but the enzyme that is deficient in this particular individual who is suffering from this particular disorder is iduronate sulfatase, and this is an X-linked disorder. Then we have MPS type 3, which is also known as Sanfilippo's syndrome, which is caused by the deficiency of one of the four enzymes, of which three are hydrolases and one is an N-acetyl transferase. In this case, there is severe mental retardation, but only mild structural changes. In your book, you can refer to the different types of MPS that we talked about here, but most of the time you will be asked about the enzymes that are deficient as well as what are the phenotypic expression of this enzyme deficiency. So you do not need to remember all the information that is given in your book, but at least remember the enzyme that is deficient. Now let us quickly look at the phenotypic features of these particular disorders. So we have MPS 1, and we already know that there is a deficiency of this particular enzyme, and you can see here there is severe mental retardation and structural changes that has happened in the patient, and this is because of the accumulation of dermatan sulfate and heparan sulfate. Then we have MPS type 2, where this particular enzyme is deficient, and this is an X-linked disorder. Again, there are severe structural and mental retardation similar to Hurler syndrome. Then we have Sanfilippo syndrome. We talked about this because of the deficiency of one of the four degradative enzymes in glycosaminoglycan metabolism, of which three are hydrolases and one is an N-acetyl transferase. In this case, there are severe mental retardation, but as you can see here in the patient, there is no phenotypic change that is observed. Then we have MPS type 4, which is known as Morquio syndrome. This happens because of galactose 6 sulfatase. There are severe structural changes in this particular individual, but these people have normal intelligence. That means there is no mental retardation happening in this kind of patients. Now we come back and quickly look at the structure of glycoproteins, one of the glycoconjugate classes that we talked about in the second lecture. Glycoproteins have two broad classes: O-linked glycoproteins, where the oligosaccharides are linked through the OH group of serine or threonine residue, and you can see here in this part of the slide, this is how a O-linked glycoprotein will look like. You do not need to remember the bonding or the chemistry behind their formation, but you of course need to remember what are the amino acids that are participating in the formation of O-linked glycoprotein. Next, we have the N-linked glycoprotein. Here, the oligosaccharide is attached by an N-glycosyl linkage, as you can see here in this particular part of the slide, to the asparagine residue of the protein. N-linked glycoprotein can be further classified into two types. One that contains high mannose, that means the oligosaccharide is consisting of mannose, and the other one is an oligosaccharide when it is composed of complex carbohydrates which contain, apart from mannose, N-acetylglucosamine, galactose, fucose, and sialic acid. Glycoproteins are synthesized on the ribosomes, which are attached to the rough endoplasmic reticulum. Dr. Nerissa will talk about this when she talks about cell biology, so I am just keeping the aspect of rough endoplasmic reticulum. From the rough endoplasmic reticulum, they are transported to the Golgi apparatus for sorting, and degradation takes place in the lysosome by hydrolytic enzymes. And if there are problems with the synthesis of these hydrolytic enzymes, we get some disorders associated with the metabolism of glycoproteins. Again, I have reiterated the function of glycoproteins because of their physiological relevance. I'm not going to read through this slide. You can see here that they have diverse functions, and till date there have been 18 different genetic diseases associated with protein impairment of protein glycosylation that are known in case of humans. Of course, these diseases, although clinically relevant, are not asked in the assessments of the board, so I have not made a list of these diseases. However, if you are interested, you can directly check them out in your textbook. Finally, we come to a discipline called glycomics, where we systematically characterize all the carbohydrate components given in a cell or tissue. And glycomics in recent times has gained popularity because it helps us to understand the mechanism of some forms of cancer. So this is some kind of a knowledge information that you need to know with regards to the aspect of glycoproteins and glycomics. As I told you, there are the degradation of glycoproteins is the lysosome is more predominantly involved in the degradation of glycoproteins, and therefore if there are problems in lysosomal enzymes, there are sometimes problems with the degradation of glycoproteins, and one such disease is called the eye cell disease. Now, the eye cell disease results from an enzyme deficiency, so that lysosomal enzyme do not acquire the targeting signal of mannose 6-phosphate. So what happens here? The fibroblasts in this disease have dense inclusion of eye bodies, and you can see here in this particular part where you can see the inclusions, and these particular cells are known as the eye cells. And the lysosomes become engorged with indigestible structures, which most of the time leads to death in infancy. And this is a particular patient who has been diagnosed with the eye cell disease. You can see here there are severe effects on the phenotype of the patient, and also there is severe mental retardation. Now I would like to quickly talk about or indicate what are some of the information that you need to read on your own. First, we need to read about blood grouping and antigens, and I'm not going to go through this particular slide. It's a very loud slide. It's actually given for your reading, so read it at your own time. It's more or less self-explanatory, and if you have difficulties or problems in this slide, we can revisit it during a revision session prior to the assessment. This is about the diseases that I talked about, and here these are actually more indicated in a more elaborate form. Again, this slide is for your own reading. Go through these slides, go through this slide in association with the other slides that I presented, where the diseases have been highlighted, and if you have any kind of problems, we can revisit this, as I said, during the revision session prior to the assessment. With that, we end this presentation, and that is also the end of the lecture series on glycogen, glycosaminoglycans, and glycoconjugates. Please sequentially listen to the lectures, and we will discuss some questions related to these lectures during the live on-site sessions, and we have a tutorial as well on this particular topic. Have a nice day, and thank you very much for listening.